Hello, RayTalksAboutCOVID here again. Welcome to my second video in a series that I'm making on the coronavirus pandemic. In the first video, I described what a virus is and what the coronavirus is in particular. In this video, I'll describe how the coronavirus gets into cells. This second video in the series will be broken down into two parts. In the first part, I'll describe the pathway by which the coronavirus gets into cells by directly passing through the plasma membrane of a cell. In the second part, I'll describe a second entry pathway that allows the coronavirus to get inside cells. This is very important because for a coronavirus to do its damage, it actually has to be able to get inside of cells. Unlike the first video where I drew as I went, here I've actually done the drawings in advance because they're a little bit more complicated and it would take too much time for me to draw as I was presenting the information to you. So let's take the first part here. Now recall from the prior video that we have the coronavirus. It has an RNA, a genetic material inside in red that's organized by the nucleocapsid protein N. And remember, I said there's three proteins in the membrane, but here I only draw one, the spike protein, which is going to end up being very important for this second video. Now, this is a cell down here with a protein known as ACE2, and this is very important protein because it's the receptor by which the coronavirus gets inside of cells. So you could think of this as a lock, and the coronavirus spike protein is the key that fits into that lock. And that's what allows the coronavirus to gain entry into a cell, just like you use a lock and key to gain entry into your house. Now, importantly, this is not drawn to scale. If you remember from my first presentation, a coronavirus, if you line them up, it would take maybe about 500 of them end to end to span the width of a cell. And I gave you the analogy that if you consider a child's bedroom in a house, let's say a 12-foot by 10-foot with an 8-foot high ceiling, the ratio of the size of the coronavirus to a cell is equivalent to the ratio of the size of this 9-volt battery to the 12 by 10 room that I just described. So the cell dwarfs the virus, but I didn't draw them to scale because then that would be too hard to see. So what's going to happen is that this coronavirus, via its spike protein, is going to dock on this ACE2 receptor of the cell, and that's going to initiate the process by which the coronavirus gets inside the cell. Okay? Now, I've only drawn one spike protein there, but remember, there's about, I think it's 60 spike proteins per individual coronavirus, any one of which can mediate this docking for the virus to get in a cell. Okay, so the first step here is we have the docking event, and the spike protein then starts going through what are known as conformational changes, and that's a fancy way of saying the spike protein changes its shape, all right? And when it changes its shape in this way, that enables an enzyme of the cell known as a protease, which is really just a form of molecular scissors that can snip proteins. So this coronavirus protein here, spike protein, has two separate pieces to it, and they're connected by a tether. And each of these pieces in the tether are all made of protein. So this molecular scissors, and I'm going to try to draw, my take on what scissors look like, snip that tether, okay, and break that tether in two, so that now this pointy piece of the coronavirus, as I've drawn it here, can reorient. And what you see is that it stays associated, it remains physically part of the spike protein, but it changes its shape. And now it's in a position where what it can do is it can actually help the virus fuse with the cell. So if you've ever played with your child with soap bubbles, for example, you know that sometimes you can make two bubbles and they come together and they don't fuse, they remain stably together. The spike protein essentially, so these are like two soap bubbles, and the spike protein essentially destabilizes this membrane so that now the membranes actually want to fuse. And that's shown here. And so when they fuse, I've just shown the start of the fusion. This widens out so the virus becomes completely incorporated into the membrane of the cell, and the red RNA of the virus enters into the cell where it can start getting copied. So that's the first entry pathway by which the virus can get inside a cell. And in the second part of this second video, I'll describe to you what the other entry pathway is.